Hello students, today we'll see the protein characterization technique. In the previous lecture we have seen different kinds of methods such as the UV method and B-synchronic method for the estimation of proteins from the given food sample. Now we'll see the next topic, that is the protein characterization. In that we'll see amino acid analysis and protein nutritional quality measurements. So in that protein nutritional quality, we will see the protein digestibility and we can say the corrected amino acid score and protein efficiency ratio. This topic we will cover today. Let us see one by one. So the first one is the amino acid analysis. So the principle. So generally amino acid analysis is used to quantitatively determine the amino acid composition of the protein. So we have to know that what kind of amino acid is protein is made up of which kind of amino acids. So this is the way, this is the motive for the analysis of the amino acid. So the protein sample is first hydrolyzed to release the amino acids. Then amino acids are then separated using the chromatography technique and then quantified. So what happens if you have to break down that protein molecule? Protein are nothing but the macromolecules. Whenever they are they are break down into smaller molecules such as amino acids, and these amino acids whenever passing through the chromatographic we can say the chromatographic column, there is a separation is going to take place, and then we have to quantify the given amino acids present in the sample. So the ion exchange chromatography, reverse phase liquid chromatography and gas chromatographic technique are used for the separation of these amino acids. This section will describe the use of ion exchange and reverse phase liquid chromatographic technique. Now, what is the procedure? So, in general, a protein sample is hydrolyzed in a constant boiling with 6 normal HCl for 24 hours to release the amino acid prior to the chromatographic separation. So here what happens, we have to hydrolyze first with the stronger acidic condition. So this is because the proteins are a very bigger molecule which is bonded with the peptide linkages, and that's why in order to analyze the amino acids we have to break down these molecules, and for that purpose we have to constantly boiling these molecules, or we can say the samples, for 24 hours. Then accurate quantification of some amino acid is very difficult because they react differently during the hydrolysis. So because as there is some kind of degradation is there while hydrolyzing the particular protein molecule, and hence the accurate quantification it is very difficult. So consequently special hydrolysis procedure must be used to prevent errors. So the tryptophan is completely destroyed by acid hydrolysis, and that's why we cannot quantify the tryptophan amount in the present sample. So the methionine, cysteine, threonine and serine are progressively destroyed during the hydrolysis. Thus, duration of hydrolysis will influence the result. So, depending upon the nature of the protein, what kind of amino acids are present, accordingly we have to apply the hydrolytic conditions, or we can say the hydrolysis. So the asparagine and glutamine are quantitatively converted into aspartic and glutamic acid respectively, and that cannot be measured. So the isoleucine and valine are hydrolyzed more slowly in the 6 normal HCl than other amino acids, while tyrosine may be oxidized. Then in general, losses of the threonine and serine can be estimated by hydrolysis of samples for 3 period of time, that is the 24-hour, 48-hour and 72 hours, followed by the amino acid analysis. Then compensation of the amino acid destruction may be made by the calculation to the zero time, assuming first order kinetics. So the valine and isoleucine are often estimated from 72 hours hydrolysis. Okay, so what happened here, from that particular hydrolysis study, what we can say, some amino acids they are free at 24 hours, some amino acids are free at 48 hours, and some amino acids they are hydrolyzed up to 72 hours. So the valines are and isoleucines are the examples which are estimated after hydrolyze up to 72 hours. Cysteine and cysteine, cysteine and cysteine can be converted to the more stable compound, that is a cystic acid, by hydrolysis in performic acid, and then hydrolyze in 6 molar HCl, and then we have to separate by chromatographic method. Tryptophan can be separated chromatographically after basic hydrolysis or analyzed using method other than the amino acid analysis. Generally, tryptophan is very prone to the acidic hydrolysis. So it is totally decomposed under acidic hydrolysis. That's why we have to perform the basic hydrolysis method or any other method than the amino acid analysis. Then what are the applications? So the amino acid analysis is used to determine the amino acid composition of protein, which determine the quantities of essential amino acids. So there are various kinds of amino acids. Some are essential, some are not essential. So we have to know what are the essential amino acids present in the sample and how much quantity is present in that particular protein. So the amino acid analysis provides the information for estimating the molecular mass of the protein. So from that we can find out the molar mass of the given bigger macromolecule, that is a protein. Now the next one is the protein nutritional quality. So the nutritional quality of a protein is determined by amino acid composition and the digestibility study of the protein. How the particular protein is digested and how much amount of outcome is getting from the digestion, we can find out the protein nutritional quality from that. So the anti-nutritional factor can affect the nutritional quality of a protein. However, foods that contain heat labile anti-nutritional factors, for example, trypsin inhibitors, are usually cooked prior to the consumption, thereby inactivating the inhibitors that might reduce the protein digestibility. Some foods contain heat stable anti-nutritional factor, for example tannin, which is heat stable anti-nutritional factor, that can be decrease the nutritive value of the protein. So the essential amino acids are those that cannot be synthesized in the body and must be present in the diet. So these are the essential amino acids. Although there are some special cases due to age and medical status of the individual, the amino acid generally categorized as essential or indispensable, which includes histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. The first limiting amino acid of the human food is defined as the essential amino acid present in the lowest amount compared to the reference protein or to the human requirements. A food scientist's concern regarding to the protein nutritional quality include meeting the requirements of nutritional labeling, formulating the products of high protein quality, and testing the effects of the food processing on the food digestibility. So the protein nutritional quality assays may utilize animal in biological test, that is in vivo, and chemical or biochemical assay, that is in vitro, that is under artificial environment we have to study, and or simply by calculations we have to find out the protein nutritional quality. Then first parameter, that is the protein digestibility. So from that we can find out the corrected amino acid score. So we can say that the protein digestibility corrected amino acid score estimates the protein nutritional quality by combining information from the calculation that compares the amount of first limiting amino acid in a protein to the amount of that amino acid in the reference protein, and an in vivo assay measuring the digestibility of the protein by the rats. So here what happened, we have to provide the particular proteins in the food to the rats, and from that digestibility what kind of changes has been made with that particular animal is measured. That means how much amount of weight is gained. We have to study that comes under the protein digestibility or corrected amino acid score. So let us see the procedure. So here, to determine the amino acid composition, first of all we have to determine the amino acid composition of the food. So calculate the amino acid score for the first limiting amino acid using the requirements of preschool age children as a reference pattern. So the amino acid score, it is equals to the mg of the amino acid in 1 gram of the test protein divided by the mg of the amino acid in 1 gram of the reference protein. So from that we can get the amino acid score, and that corrected amino acid score, it is nothing but the protein digestibility. So the feed male winnerling rats standardized diets with 10% test protein or with no protein following the procedure for 2 protein digestibility. So here what happened, we have to take the particular protein diet. So we have to take the winnerling rats, that is the particular rat species, that is winnerling rats, we have to consider, and we have to provide the diets with 10% test protein. So what is that, we have already find out the amino acid score. So that test protein is 10% is provided to the rats. Okay, and there are some rats which are having no protein source with them, means what, we have not provided the protein to the some ones. Okay, we can call it as a standard, or we can say the blank. Okay, and then following the procedure we have to see what kind of changes is made, and that is a this method is mentioned in the AOAC, that is a AOAC method 991.29. So, true digestibility, it is calculated based on the nitrogen ingested and the feed intake, corrected for metabolic losses in the phases. If available, published values of the true digestibility of the test protein can be used, and from that we have to calculate the protein digestibility corrected amino acid score, and which is given as amino acid score into percentage of true digestibility, and true digestibility is calculated by nitrogen ingested and feed intake corrected for the metabolic losses in the phases. So for the nutritional labeling, 50 gram daily value of the protein, it is provided. So the percentage of daily value equals to 100 into gram of protein serving into PDCAAS, that is protein digestibility corrected amino acid score divided by the 50 gram of protein. So this is the percentage daily value. Then what are the applications of protein digestibility corrected amino acid score? So the Nutritional Labeling and Education Act requires that percent daily value used on the nutritional label must be determined using protein digestibility corrected amino acid score method, except for the foods which are intended for consumption by the infants, that is newly born childs. So because of the time and the cost associated with the protein digestibility corrected amino acid score method, protein on the nutritional label is usually expressed only as amount and not the percent of the daily value. However, if food label includes any claim regarding to the protein, the nutrition label must include protein expressed as the percent of the daily value. So the PDAS method generally is thought to better estimate the protein quality for the humans than the protein efficiency ratio method, which measures the rat growth. So the rat growth is not comparable to the adult humans, but it is more comparable to that of the human infants. Therefore, the PER, protein efficiency ratio method, is used to estimate protein quality of only infant foods. Now the next one is the protein efficiency ratio. So what is the principle? So the protein efficiency ratio is estimates the protein nutritional quality in an in vivo assay by measuring the rat growth as a weight gain per gram of the protein feed. So procedure. So determine the nitrogen content of the test protein containing sample and calculate the protein content. So first we have to formulate a standardized test protein diet and casein control diet to each containing 10% protein. So similarly what we have to do, we have to provide the 10% of the given protein diet. Then feed groups of the male vinailing rats the diet and water for 28 days. Record the weight of each animal at the beginning of the assay at least every 7 days. We have to have the 7 days duration to take a weight of each uh, rats during the assay, and at the end of the 28 days means what we have to perform the 4 cycles for the taking the weights. So the record the food intake of each animal during the 28 days of feeding trial, and from that we can calculate the protein efficiency ratio. So they calculate the protein efficiency ratio using the average total weight gain and average total protein intake for each diet group at day 28. So the protein efficiency ratio, it is nothing but the total weight gain of test group in gram divided by the total protein consumed in gram by the particular rats. So normalize the protein efficiency ratio value for the test protein, that is it compares the quality of the test protein to that of the casein by assigning the casein as a protein efficiency ratio of the 2.5. So adjusted or corrected protein efficiency ratio, it is equals to the PER, that is the protein efficiency ratio of the test protein divided by the protein efficiency ratio of the casein as a control. Now what are the applications? So the Food and Drug Administration requires that the PER method, protein efficiency ratio method, be used when determining protein as a percent of the daily value on the nutritional level of the foods which are intended for the consumption by the infants. The PER method is limited to this application because the essential amino acid requirements for the young rats are similar to those of the human infants, but not to those of the other age groups. The PR method is time consuming and it does not give any value to the protein for its ability to simplify maintain body weight. That is, the protein that produces no weight gain in the assay has PER is always 0. Okay, so this is all about the protein nutritional qualities. In the next lecture we will see other characterization methods. Okay, thank you.